So I want you to just take a second and think about the last time you tried to learn a new physical skill.
Oh, like picking up a new sport or something.
Yeah, exactly. Like learning to ride a bike or maybe trying to perfect your backhand swing in tennis. You try the movement, you fumble, you try it again.
Right, and it feels super frustrating at first.
It really does. But somehow, eventually, your brain just figures it out. It knows the exact difference between a good movement that you want to keep and a mistake that needs fixing.
And the wild thing is, it feels entirely automatic to us on a conscious level. I mean, you hit the tennis ball into the net, and your brain instantly recalibrates the muscle tension and the timing for your next swing.
But if you zoom in, like at a cellular level, that recalibration should actually be a logistical nightmare.
Well, absolutely. A total nightmare.
Because the brain is just this cacophony of electrical noise, right?
Mm-hmm.
With billions of neurons firing constantly. So how on earth does your motor command center actually filter out all that chaotic daily static to capture the exact microscopic moment a learning update is needed?
That is exactly the question we are exploring today.
Yes, and we have a truly fascinating source to guide us. We are taking a deep dive into a groundbreaking 2026 paper published in Nature Neuroscience. It's titled Synchronous Climbing Fiber Activity Enables Instructive Signaling for Cerebellar Learning Through Modulation of Disinhibitory Circuits.
It's a bit of a mouthful, but our setting for this deep dive is the cerebellum, which is that dense, tightly packed structure at the base of your brain that acts as your motor control center.
Right, the little brain.
Exactly. And what this research does is it actually solves a neuroscience mystery that has baffled the field for decades.
A literal decades-old mystery.
Yeah. And the answer to how we learn physical skills involves this microscopic game of telephone and a highly counterintuitive mechanism known as serial disinhibition.
Okay, let's unpack the cast of characters here first, because to understand this mystery, we really need to know who is operating inside this microscopic world.
We definitely need a roster.
Right. So there are three main players in the cerebellum. First, we have the Purkinje cells, or PCs. And I kind of like to think of them as the VIPs of the motor center.
That's a great way to look at it. They're these massive, highly complex neurons with branching structures that honestly look like ancient oak trees.
Yeah, they're huge.
Yeah.
And they are the main output neurons. So ultimately, every single motor command and every learning update flows through them.
They are the final common pathway for the cerebellar cortex. Basically, if you want to change a physical behavior, you have to change the synaptic connections on those Purkinje cells.
Okay, so player number two, we have the climbing fibers, or CFs. And these have traditionally been viewed as, the teachers.
Right, the ones giving the instruction.
Yeah, classic neuroscience theories going all the way back to the Mar Albacete models from the, I think this is 1960s and 70s.
Yep, the 60s and 70s.
They established this very clean, logical framework. David Marr, James Alvis, and Masao Ito proposed that climbing fibers send a distinct error signal from a deeper brain structure called the inferior olive right up into the Purkinje cells to trigger learning.
It's a very simple cause and effect. You make a mistake, the climbing fiber zaps the Purkinje cell, and the cell adapts.
Simple, elegant. But then we have the third players, which are the molecular layer interneurons, or MLIs, and these act as the bouncers of the network.
The bouncers, yeah. I love that analogy.
Because they provide this incredibly powerful localized inhibition to the Purkinje cells. Their entire job is to keep the VIPs quiet, essentially stopping the Purkinje cells from changing their connections wildly every time there's a stray electrical signal.
Which is super important for stability. But that brings us to the central paradox of this entire paper.
Yes, the plot twist.
Right. That classic textbook model where the climbing fiber only fires when you make a mistake. Well, it turns out to be fundamentally flawed.
Wait, really? Flawed how?
Decades of functional recordings in live animals have shown us that climbing fibers don't just fire when there is an error, they actually discharge persistently.
Like all the time?
All the time. They fire across all kinds of behaviors, even when you execute a movement flawlessly.
Okay, so it is known as the continuous alarm paradox. The signal that is supposed to be this strict, highly specific teacher is actually just firing constantly in the background.
Exactly.
But hang on, that makes zero sense from a biological perspective.
Yeah.
I mean, the human brain is notoriously energy efficient, right?
Highly efficient, yes.
Firing an action potential takes massive biological energy.
Yeah.
Why would evolution build a smirk alarm that constantly goes off every few seconds just because the battery is in?
It's a great question.
Because if the alarm is always ringing, the Purkinje cell should be constantly triggering maladaptive learning. Like we would completely ruin our physical coordination just by walking down the street.
And that exact contradiction is what researchers have debated for years. I mean, if the climbing fiber signal is continuous and seemingly indiscriminate, the actual instructive value, the command to learn, cannot possibly be contained in the raw signal itself.
Right, because if it was, we'd be constantly unlearning how to walk.
Precisely. So what this paper hypothesized and went to incredible lengths to prove is that the answer lies in how the surrounding bouncers, that inhibitory MLI network, how they conditionally filter those continuous signals.
Okay, so just to pause and do a quick recap of where we are so far to make sure I'm cracking with the logic here. We have a continuously ringing alarm and VIPs that only need to be worn during an actual fire.
Spot on.
And the hypothesis is that the network of bouncers is somehow decoding this alarm. But to figure out how they decode it, you can't just look at a functional MRI or, broad electrical signals, right?
No, not at all. That won't give you the resolution you need.
You have to trace the actual physical wiring between these specific cells.
Right. You have to look at the connectomics. You really have to see the physical architecture of the circuit at a microscopic level, which requires mapping tens of thousands of individual connections.
And if you're listening to this and wondering how scientists can possibly map microscopic connections between individual brain cells without losing their minds.
It takes a lot of patience.
The technology they used here is just fascinating. It's a technique called serial block face electron microscopy.
It is a truly remarkable process. So they took a sample of a very specific region in a mouse's cerebellum called lobule IVV.
Lobule IVV?
Yeah.
Oh.
Think of lobule IVV as a specific zip code in the brain dedicated to processing particular sensory and motor information. They embedded this tiny piece of tissue in resin and then used a microscopic diamond knife to slice it incredibly thin.
And we're talking nanometers thin here. I mean, they slice it, an electron beam scans the surface to take an ultra-high-resolution picture, and then they slice it again.
And they repeat this thousands of times.
Thousands of times to build this massive three-dimensional digital puzzle of the molecular layer. And then they use advanced machine learning algorithms to trace the chaotic tangle of wires.
Right, combined with extensive manual proofreading by human researchers, because they have to ensure every single branch is correct.
Which is just mind-blowing. The sheer scale of what they reconstructed is staggering. Inside a microscopic cube of tissue measuring just 175 by 122 by 50 micrometers.
Which is smaller than a grain of salt, by the way.
Right. They successfully mapped 219,774 individual synapses. They completely traced the local circuits, including nearly 34,000 parallel fibers.
It's unprecedented. In typical neuroscience imaging, you usually get a blurry picture of a forest. Here, they mapped every single leaf and twig on the trees.
And because they had that level of structural detail, they found something that had been completely obscured before. Our bouncers, the MLIs, they're not just a single monolithic group of cells doing the same job.
No, they aren't. What's fascinating here is, by mapping the exact connectivity, they discovered the MLIs actually segregate into two highly distinct functional subtypes.
MLI1 and MLI2.
Exactly. And this changes the entire architecture of the circuit. MLI1s are your classic bouncers. They directly wire into the Purkinje cells to inhibit them.
Keeping the VIPs quiet.
Right, but MLI2s are entirely different. They avoid the VIPs completely. Instead, they selectively wire into the MLI1s.
Wait, so they inhibit the inhibitors?
Yes. They are essentially bouncers for the bouncers.
Oh, wow. Which introduces a completely hidden layer of control, but finding MLI2 wasn't enough to solve the paradox.
Right, because they still needed to know how the climbing fibers are constantly ringing alarms interacted with these specific bouncers.
Right, so they combed through that massive 3D data set looking for a needle in a haystack. And out of nearly 1,000 close physical contacts, they confirmed 53 direct structural synapses where the climbing fibers bypass everything else and plug directly into the MLI2s.
It's just 53. It is a rare targeted connection. The climbing fibers form these structures called planar arbors.
Planar arbors, okay.
Yeah, think of them as flattened branching trees that kind of snake through the tissue. They mostly avoid normal synapses, but they deliberately seek out and connect to MLI2 cells.
So this establishes a parallel signaling pathway. The climbing fiber isn't just sending signals toward the Purkinje cell. It is simultaneously communicating directly with the bouncer that controls the other bouncer.
Exactly.
Okay, let me visualize this to make sure the mechanics make sense.
Yeah.
Because we know that in neurobiology, inhibiting an inhibitor creates excitation, right?
Yes, it's a double negative.
Right. This is what the paper calls serial disinhibition. So if our VIP, the Purkinje cell, is being held back from learning by the first bouncer, MLI1.
Right.
But then a second bouncer, MLI2, receives a signal from the alarm, steps in, and intercepts MLI1.
And puts him in a headlock, basically.
Exactly. Because MLI1 is now tied up, our VIP is completely unbothered. The inhibition is lifted, and the Purkinje cell is free to receive new instructions and party.
That perfectly describes the mechanism of serial disinhibition. When MLI2 activates, it puts the brakes on MLI1, which releases the brakes on the Purkinje cell.
But wait, here's where it gets really interesting. If MLI2 is always capable of shutting down MLI1, and the climbing fibers are constantly firing their alarms in the background, why isn't the Purkinje cell just constantly, completely uninhibited?
That is the big question.
Because we are right back to the original paradox. Why doesn't MLI2 just permanently intercept MLI1 and leave the system in a constant state of learning? There has to be a catch.
There is a catch, and this is really the absolute crux of the paper. This is how the brain actively filters the noise.
Okay, lay it on me.
Through computational modeling, which was constrained by their new anatomical data, the researchers discovered that MLI2s require convergent inputs to activate.
Convergent inputs?
Yes. A single climbing fiber firing that regular, persistent background beeping of the alarm when you're just moving normally is simply too weak. It doesn't depolarize the MLI2 cell enough to trigger an action potential.
Oh, I see. So if I can use a visual memory aid here, a single background signal is like a single drop of water hitting a dam. The network easily sheds it, it ignores it completely.
That's a great way to picture it. But during a real motor error, like when you actually swing and miss that tennis ball, something special happens down in the inferior olive where these signals originate.
What happens?
The neurons down there are physically connected to each other by structures called gap junctions.
Okay, gap junctions. Those are literal physical tunnels connecting the membranes of adjacent cells, right?
Yes, exactly. They allow electrical currents to pass directly between the cells. And because of these physical tunnels, when a true error occurs, the olivary neurons are forced to fire synchronously.
Meaning a whole population of climbing fibers fires at the exact same millisecond.
Precisely.
So it's not a single drop of water anymore. It's a synchronized tidal wave hitting the dam.
Yes. Because the MLI2s integrate inputs from multiple climbing fibers simultaneously, this synchronous tidal wave is what strongly activates them. The population synchrony is the key that turns the lock.
Wow.
Yeah. When that synchronous wave hits, MLI2 fully activates, intercepts MLI1, and drops the inhibition on the Purkinje cell.
Okay, so let's do another quick recap. The continuous background firing is just asynchronous, scattered drops of water, so it gets completely filtered out by the MLI network.
Right.
But the true error signal is synchronous, a tidal wave that breaks through the inhibitory gate. So when that inhibition drops, what actually happens inside the Purkinje cell that equals learning?
The freed Purkinje cell experiences a massive influx of calcium into its dendrites.
Calcium.
Yeah. In cellular biology, a sudden spike in intracellular calcium is the biological trigger for synaptic plasticity. It causes the cell to internalize certain receptors, physically changing the connection weights.
So it is the molecular manifestation of the cell saying, that movement was wrong, adjust the circuit for next time.
Exactly. So the meaning isn't in the signal itself. The instructive command to learn emerges entirely from the timing of the crowd.
That is just brilliant biological engineering.
It really fundamentally shifts the conceptual framework of cerebellar learning. The field has spent decades trying to decode the message carried by a single wire. This shows us that the instructive value is a circuit-level computation based on timing.
But, as with everything in neuroscience, structural maps and computational models are just highly educated theories until you can prove them in a living, breathing animal.
That's very true.
To truly prove this, you'd have to go into a live brain and surgically remove that specific disinhibitory pathway without damaging the rest of the motor system and see if the animal loses its ability to learn.
And achieving that required an incredibly sophisticated experiment. They used a highly targeted molecular genetic approach to essentially erase just the connection between the two bouncers.
Okay, let's break down the tools they use because it honestly sounds like science fiction. They started with what are called kit-cree mice.
Right, kit-cree mice.
From my understanding, these mice are genetically engineered so that researchers can target very specific cellular zip codes. In this case, just the MLI interneurons in a region of the cerebellum called the flocculus.
And the flocculus is basically the control room for eye movements.
Okay. Then they used an adeno-associated virus, or AAV. An AAV is essentially a harmless viral delivery truck, right?
Yeah, it just delivers a payload.
Right. They loaded this truck with a specialized payload called GFE3 and injected it into that exact zip code.
And GFE3 is where the magic happens. It's an E3 ligase targeted at a structural protein called gefrin.
Okay, rather than throwing jargon around, let's look at its actual function. An E3 ligase is basically a cellular garbage disposal tag, right? And gefrin is the structural glue that holds inhibitory synapses together.
That's a perfect description.
So GFE3 acts as a customized microscopic assassin. It was genetically programmed to hunt down and dissolve the specific protein glue holding the MLI synapses together.
Exactly. By dissolving that glue, the virus selectively destroyed the connection from MLI2 to MLI1. It effectively removed the second bouncer's ability to intercept the first bouncer. But crucially, because of how they targeted it, it left the direct inhibition from the first bouncer to the Purkinje cell completely intact.
So the VIP is still being held back by the first bouncer, but the mechanism to free the VIP, that tidal wave pathway, is entirely gone.
Right, it's been physically erased.
The alarms are still ringing, the engine is still intact, but the single gear that translates the synchronous alarm into action has been removed.
So what does this all mean? Like, what actually happened to the mice when they put them to the test?
Well, they put the mice through a classic motor learning behavioral test involving the vestibulo-ocular reflex, or VOR.
Okay, the VOR is that reflex that keeps your eyes locked on a target even when your head is turning. Like, if you turn your head to the left, your eyes automatically rotate to the right at the exact same speed to keep the image stable on your retina.
Exactly. And researchers can artificially induce an error in this system. They essentially trick the brain into thinking its eye movements are slightly off, which forces the mouse's climbing fibers to signal an error to teach the mouse to adapt its reflex.
Right. And in normal, healthy mice, the synchronous climbing fibers fired, the disinhibitory pathway opened up, the calcium spiked in the Purkinje cells, and the mice smoothly learned the new reflex.
And the mice with the microscopic assassins, the ones missing the MLI2 connection.
They completely failed at this climbing fiber-instructed motor learning.
They just couldn't learn it.
No, they couldn't. The climbing fibers were still firing perfectly fine. The visual errors were being accurately registered by the brain.
But because that highly specific MLI to MLI inhibition was broken, the synchrony key had nowhere to turn.
Exactly. The MLI1 bouncer never got intercepted, the Purkinje cell remained stubbornly inhibited, the calcium never spiked, and the learning was entirely locked out.
That is just incredible. It is the definitive behavioral proof. The necessity of this hidden disinhibitory pathway is absolute.
It proves, in a living system, that instructive signaling is not a single input. It is the product of network-level processing.
So taking a step back from the microscopic mechanisms, the diamond knives, the viral delivery trucks, the VIPs and bouncers, the grand takeaway here is really profound.
It really is.
We have to stop viewing neural learning as a simple feedback loop where one wire carries a distinct mistake message.
Right, because this paper proves that the actual knowledge of a mistake is an emergent property of population synchrony.
The critical information isn't held in an individual data point. It emerges entirely from how the network dynamically responds to the rhythm of the population.
It is an exquisite balance of inhibition and disinhibition, beautifully tuned by the timing of our neural populations to filter out the noise of our daily lives.
It really makes you look at your own brain differently. I mean, as you, the listener, go about your day-to-day, picking up your coffee cup, typing on your keyboard, stepping off a curb, think about how elegant this biological design is.
It's constantly working in the background.
Yeah. Your cerebellum is constantly buzzing with chaotic electrical static, but it is seamlessly using these complex, hidden network dynamics to shed the noise, only capturing the synchronized tidal waves that truly matter for your adaptation and survival.
It is a level of biological efficiency that we are honestly only just beginning to truly map and understand.
Then I want to leave you with a final provocative thought to mull over, especially given the technological era we find ourselves in today.
Oh, that's a good one.
We are currently obsessed with building artificial intelligence. We pump billions of individual isolated data points into these massive artificial neural networks, trying to teach them to learn and adapt.
Right.
But if our own brain's supercomputer, the cerebellum, arguably the most efficient learning machine on Earth, if it relies fundamentally on population synchrony and specialized disinhibitory circuits to decide what information is actually worth learning, what does that teach us about how we design our AI?
It's a paradigm shift.
Are we spending too much time trying to optimize the individual data points and completely missing the sheer computational power of the network's rhythm?
That is a fantastic question to carry forward, and one that computational neuroscientists are likely asking themselves right now.
Something to think about the next time you try to perfect that tennis swing. Thanks for joining us on this exploration of the mind's hidden machinery. Keep wondering, keep learning, and we'll catch you on the next Deep Dive.